This is part 2 of our lecture on DNA. We're going on to see how DNA replicates itself, which is a somewhat complex process. I will remind you that there are also some really nice animations I've made for you, made available for you in the online portion of the classroom. These will really help you visualize and understand this process. So be sure you watch those not once or twice, but several times, and take notes until you feel like you are comfortable with understanding this process. Okay, so here we go into DNA replication. Now, when we think about DNA replication in its most basic, we think of it as a process that is what we call semiconservative. And what that simply means is that because DNA is a double-stranded molecule, we know that during replication, one of the two parent strands or original strands goes to one of the new daughter strands, and the other parent strand goes to the other daughter molecule. Okay, so that means that if here's my parent strand, one of those two strands, one of the parent strands goes to becomes one half of the daughter strand, and then a new strand is synthesized to pair with it. And for the other daughter strand, one of the parent strands goes with it, and a new daughter strand that is complementary is synthesized to pair with it. So we half conserve the parent strand in each daughter strand. So how does this process take place? Well, there are kind of three basic phases or stages to replication. The first of which being unwinding. Remember again that DNA is a double-stranded helix. Because it is twisted, the proteins that are necessary to help with the process of copying the DNA cannot physically get into the DNA molecule to do the synthesizing of new strands until that strand is unwound and opened up so there's room for those proteins. So there is a small protein called helicase. See how it ends in ASE? We know it's an enzyme then. Helicase basically unwinds the DNA. Okay, it also produces a little nick or a cut in one of the two strands. We'll get to that later, or you'll see that in animation. But the big job that helicase does is it unwinds the DNA, and it also helps to break apart the hydrogen bonds so that we split apart the molecule, as you can see here, to form two separate strands. The next part, or the next phase of DNA replication, is complementary base pairing. And this is where an enzyme called DNA polymerase, again it ends in ASE so you know it's an enzyme, DNA polymerase makes a new polymer of DNA. Remember, a polymer is a long chain of organic molecules strung together, in this case a long chain of nucleic acids. And it does it by, for example, if we look on this particular strand, we have a G here, so we would pair that with a C. We have a G here, so we would pair that with a C. Here we have a T, so an A has been paired with it. Here we have a T, so we have an A paired with it. So basically, what the DNA polymerase does is it reads the sequence of nucleotides on the parent strand, and it brings in free-floating nucleotides to pair up appropriately with the parent strand through complementary base pairing. Again, that idea that A's always pair with T's, C's always pair with G's. Now, while that's happening on one strand, the same thing's occurring on the other strand. But because the DNA strands are anti-parallel, one goes 5' to 3' and the other goes 3' to 5', the direction in which synthesis occurs is going in the opposite direction. And this creates something called leading and lagging strands. And I'm going to hold off until the next slide to actually talk about the difference between those two. The one thing to remember with all of this complementary base pairing that's going on here and the DNA polymerase is that we've got to have nucleotides, free-floating nucleotides, free-floating C's, free-floating G's, free-floating A's, free-floating T's, floating around that DNA polymerase can then grab and then pair up with the existing strands sequence of nucleotides so that we reform a double helix in the daughter strand. This process of base pairing, of synthesizing, always moves in the 5' to 3' direction when you're talking about the new strand that's being made. Okay, the new strand is always synthesized starting at the new strand's 5' end and moving toward the new strand's 3' end. The last step of DNA replication is joining. Now that DNA polymerase has brought in and paired up all these A's, C's, T's, and G's, we have to actually link them together. Now the hydrogen bondings already occurred down the middle between the A's and the T's, the C's, and the G's, but along the phosphate sugar backbone, we have not yet joined the nucleotides, and that's what the enzyme ligase does. It literally glues together the newly brought in nucleotides one with another. Now, I'm going to talk very briefly about leading and lagging strands. I cannot emphasize enough to you how important it is you watch the animations related to this. It's difficult to describe this using a static picture, and I will spend time in class helping you better understand this process. We'll watch some of the same animations again, maybe a few different ones, and we'll also go through it together as a class so you understand it better. But do not just rely on what I'm going to show you here. You need to pay attention in class on the day I discuss this, and you also need to take advantage of those animations I've made available for you online. That being said, when we look at the two strands of DNA that have been separated, here's the helicase that separates them apart. Remember, helicase unwinds the DNA and splits apart the hydrogen bonding, opens it up. You can see that the blue strand here is the parent strand. The blue strand is the parent strand. And you will see this is the parent strand that ends in a 3'. Remember, I told you that the new daughter strand is always synthesized starting at the new daughter strand's 5' end and moving to the new daughter strand's 3' end. So here is the new daughter strand. Indeed, we've started at a 5' end and we're moving this way towards the new daughter strand's 3' end. This strand is called the leading strand, and here's why. Replication of the DNA occurs fastest on this strand. You'll see here that the DNA polymerase, which is in charge of bringing in new nucleotides to match up with the parent strand, can just keep moving towards where helicase is unwinding the parent DNA. Helicase just keeps moving in this direction and unwinding the DNA. And this place here where we get the V, where we get the two parent strands split apart, is called the replication fork. So in this case, DNA polymerase on the leading strand can simply keep moving towards the replication fork as helicase continues to unwind more DNA. So it can just continuously synthesize a new copy of DNA. Things are a bit more complicated on the lagging strand. Remember, I told you that DNA is anti-parallel. So if we have the parent 3' end here, that means that for the parent strand on this side, we end in the 5' end. That complicates things because remember, DNA synthesis has to begin at the new strand's 5' end and move toward the newly synthesized strand's 3' end, which means in this case down here we have to be moving this way as we synthesize. The problem with that is that you will notice as DNA polymerase here hooks onto the parent DNA and begins synthesizing in this direction, it is moving away from the replication fork. Meanwhile, helicase continues to unwind, and then we have a big old space here where there is no DNA polymerase attaching, and thus no copying of this DNA going on. So what happens is every once in a while, as helicase unwinds a bit more DNA, a new strand of DNA polymerase pops in and begins to replicate DNA on the lagging strand. And so you can see here, here's one DNA polymerase, here's another molecule of DNA polymerase, here's another molecule of DNA polymerase, and they are all making little fragments of new DNA. These little fragments of DNA are called Okazaki fragments. And they're called Okazaki fragments because a guy by the name of Okazaki, last name Okazaki, was the one that first described these. So we get these little Okazaki fragments that later have to be joined together one to another to make a continuous strand, and DNA ligase helps to do that. But because we're making a bunch of little fragments that later have to be joined together, this process takes longer than the process of copying DNA on the leading strand. And because the process takes longer, we call this the lagging strand. Literally, the synthesis of DNA lags behind the synthesis on the leading strand. Now, there's one important point that I have not yet mentioned here, and I want to back up to the previous slide and mention this. Before DNA polymerase can begin to do its complementary base pairing, something special has to occur. DNA polymerase cannot attach directly to DNA. I'm going to say that again. DNA polymerase cannot attach directly to DNA. Instead, DNA polymerase must first attach to a small sequence of RNA, and then it can begin to synthesize DNA. If you look in this picture here, you will see that at the beginning of this Okazaki fragment, there's a little RNA primer. There is another molecule, another type of DNA polymerase that will first synthesize an RNA primer before the DNA polymerase that we've talked about is able to attach. So then once we have an RNA primer, our DNA primase attaches, and then we can start to get synthesis. But if I don't have an RNA primer first attached to the parent strand, there's no way for that DNA polymerase to attach. Phew, that's enough on that. Let's move on. In our next part, we're going to talk a little bit about mutations.